So we looked at glucose breakdown, and now we're going to look at, so we went through glycolysis, and we started going into the citric acid cycle, but we're going to look at it a little bit more in depth. And so, just as a review, each Acetyl-CoA that's produced will provide 8 electrons for the electron transport chain. So every NADH carries 2 electrons, so it carries an electron pair. And every FADH2 can either carry 1 or 2 electrons, but for our purposes we're putting 2 electrons as what it's carrying. Now any NADH produced in the cytosol is transported by the malate aspartate shuttle. And it's taken into the mitochondria. And then the source of acetyl-CoA, so I'm going to move some stuff around. The source of acetyl-CoA is in the well-fed state, it's primarily pyruvate. So pyruvate combines with coenzyme A to form acetyl-CoA, and that's done by pyruvate dehydrogenase, and so PDH, pyruvate dehydrogenase. So that's in the well-fed state. Now in the starvation state, and in the well-fed state, most of pyruvate comes from glycolysis, but in the starvation state, most pyruvate comes from the amino acid skeletons. And we're going to look at that a little bit more later. So this gives us acetyl-CoA, and this is primarily from the well-fed state. And fatty acids give us acetyl-CoA in the starvation state. Starvation. ON. Now this is the citric acid cycle, or the TriCarboxylic Acid Cycle, and it gets its name from the first 4 intermediates. So, well, you see that citrate has 1, 2, 3 carboxylic acids, and these are actually the anions of a carboxylic acid. These are carboxylate anions, and they're going to be anions in a physiological pH, but just know that they come from carboxylic acids. So citrate, and then isocitrate and oxalosuccinate, and then of course cis. And I'm not sure exactly how it's supposed to be pronounced, but aconitate is how I'm pronouncing it. So cis-aconitate. And the typical flow from acetyl-CoA is that acetyl-CoA will combine with oxaloacetate, and coenzyme A will be released, and citrate will be formed. And then aconitase acts on citrate to form cis-aconitate, and then the same enzyme performs the second step of this 2-step reaction to form isocitrate. So aconitase acts twice, doing a slightly different thing each time. First time it dehydrates, the second time it hydrates. So basically it's shuffling around water. And so you can see this alcohol group, and one of these hydrogens is taken off from over here. So the alcohol group is missing, and we have a CH1 instead of a CH2. And then that hydrogen, so where the alcohol was, the hydrogen gets placed, and where the hydrogen was, the alcohol gets placed. And so that forms isocitrate. Then isocitrate dehydrogenase forms 1 NADH and creates oxalosuccinate, and then the same enzyme reacts again, removing a carbon dioxide, and I'm going to move this up, and then so it removes the carbon dioxide and forms alpha-ketoglutarate. Now you may remember from a past video that alpha-ketoglutarate can be made from glutamate. That's an I. Maybe it's an A, I don't remember. Yeah, I think it's an A. So, and then alpha-ketoglutarate gets acted on by its dehydrogenase enzyme, and forms another NADH and produces a succinyl-CoA. And succinyl-CoA synthetase forms succinate and produces a GTP, which is energetically basically equivalent to an ATP. And then succinate gets acted on by succinate dehydrogenase, forming an FADH2 and fumarate. Then fumarase hydrates to L-malate, and then L-malate gets acted on by a malate dehydrogenase, forming another NADH and reforming the starting molecule oxaloacetate so that it can recombine with another acetyl-CoA as it comes in. So one thing that's important to kind of keep in mind is that the coenzyme A is removed and it can come back up here and react with another pyruvate to reform another acetyl-CoA. And so the coenzyme A gets recycled. And then another kind of important thing to keep in mind is the number of NADHs. So you have an NADH here, another one here. You get a GTP here, an FADH2 here, and then if I pull this down a little bit, I get my last NADH right here. So NADH, we get 3, and FADH2, we get 1, and we get a GTP/ATP equivalent of 1. So if this carries 2 electrons each, then I have 6 electrons there, 2 electrons from there. So the TCA cycle provides me with 8 high-energy electrons to feed into the electron transport chain. Now I like this particular image because it shows a couple things. It shows the structure of each of the molecules to some extent, and it also shows in clear detail where the same enzymes are being used twice. So it's not going to omit this intermediate step. It's not going to omit this intermediate step, like in so many of the images that you can find in textbooks and on the internet. However, what I don't like is it doesn't highlight the irreversible steps. So here in this image, you actually see that pyruvate dehydrogenase, so from pyruvate to acetyl-CoA, is the first irreversible reaction, so to speak. So under physiologic conditions, this enzyme will not catalyze a backwards reaction. And so then the second step, being the citrate synthase, is irreversible. Then the 2 reactions of isocitrate dehydrogenase to go from isocitrate to oxalosuccinate to alpha-ketoglutarate is irreversible. And then from using the alpha-ketoglutarate dehydrogenase to produce succinyl-CoA is also irreversible. So in each of these steps, you can see that having a high amount of ATP will usually inhibit all of these enzymes. And so that's also right here and right here. So being in a high energy state will inhibit that. And then you can also see, like here, the succinyl-CoA. If we have a lot of succinyl-CoA, that also inhibits the reaction, as well as here having NADH. But the isocitrate dehydrogenase can be upregulated, as well as the alpha-ketoglutarate dehydrogenase by calcium, and especially in skeletal muscles, this is important to couple the contraction of the muscle with the production of energy. And so you can also see that calcium acts on pyruvate dehydrogenase as well. And so we also see that pyruvate dehydrogenase will be inhibited by fatty acids. So in fatty acids, so if we're using fats to produce the electron equivalents for the ETS, then we can use pyruvate for something else such as gluconeogenesis, and that will inhibit this enzyme. Now the last thing to take note of is this reaction right here. So from succinate to fumarate, if you remember right, you get the production of FADH2 DH2, and I showed that on the previous picture. Why that's interesting is because succinate dehydrogenase is part of something called complex 2. Complex 2 is an enzyme in the electron transport chain. So succinate is converted to fumarate by an enzyme that's in the electron transport chain, and it actively produces electrons for the electron transport chain. So this is kind of showing you that all of this is happening very close. The action takes place very close to where it needs to take place. Okay, in the next video we're going to look at so we only looked at where pyruvate and acetyl-CoA feed into the citric acid cycle. The next video we're going to look at other metabolic intermediates and where they can flow into the citric acid cycle from different points and how that affects things. And we'll also look at using different intermediates from the citric acid cycle for the biosynthesis of other molecules rather than for the production of CO2 alone. The main take-home point is that the TCA is used to provide electron equivalents for the electron transport chain.